As promised, this is a short podcast talking about how we map genes using bacterial conjugation. In class we talked about it, but it was a poor example and a poor representation on that image that was just really hard to read. So I thought it'd be nice if I just wrote it out here. Also, there's a problem in your homework that deals with this too, and there's a short video that goes with that, and I'd encourage you to watch that. What I want to begin with is by talking about the different strains we're going to use. And this first strain is going to be an HFR strain, high frequency of recombination. And let's draw it like so. And on the chromosome here, I'm going to draw it quite large, just so we can draw things on it. And we're going to have a couple genes. We're just going to show 3 genes. The example in class had a whole bunch of genes, and I think that added to the complexity. So let's just talk about these 3 genes. And let's just make up something here. I'm going to put M, just because I don't have room to write the whole thing out, but this is for Met plus, so M plus. And the next gene up here is L plus, standing for Lu plus. And the next one is U for Ura plus. These cells can make their own methionine, leucine, and uracil. Now, because they're HFR, they can also go through conjugation because they have this down here, the F factor. That used to be a plasmid, but it's right here. And this is what's going to allow it to later on undergo conjugation. It codes the genes for the sex pillars. Now let's write our other strain over here. And this strain is an F- strain. It can't undergo conjugation unless you mix it with a cell, like an HFR cell or an F plus cell. Let's draw this cell now. And I'm going to draw the chromosome in it. Again, I'll draw it large, like so. And it's going to be Met minus, so I'm going to make it a lowercase m. And put a minus by it. Then there's a leucine minus, so I'm going to make a lowercase l and a minus. And the next gene, remember, is the uracil gene. And I'm going to put a U minus. This cell cannot make its own methionine, leucine, or uracil. All right, let's begin by asking what these cells can do on their own. Oh, one more thing. We have to put it here. This particular cell here is, just write it here, is strep sensitive, meaning it cannot grow on media containing streptomycin, the antibiotic. Streptomycin will kill this cell. This cell over here has the gene for strep resistance, strep R. You grow this in media containing streptomycin, this F- strain will grow. Let's talk about minimal media first. I'm going to abbreviate that, just MINMED, minimal media. This cell, if you place it on minimal media, it will grow. So I'm going to put a plus here, meaning it will grow. Just to remind yourself, minimal media does not contain any of the amino acids or nucleic acids like methionine, leucine, or uracil. So if a cell grows on this, if a bacterial cell grows on minimal media, that means it has the ability to make all of these nutrients. And then let's talk about complete media. And I'm just going to go COM and complete media. In your notes, what you want to say is complete media has all the nutrients that a cell needs to survive. It has the uracil, it has the leucine, it has the methionine, plus all the other amino acids and nutrients. Now, this cell will grow on it. It can make its own, but if the media is going to supply it, all the better. And then what I'm going to put here is complete media plus streptomycin. And I'm just going to put ST. So this media contains the antibiotic streptomycin. This is sensitive to it, so it will not grow. I'm going to talk about some other medias too, and I apologize, I had to scratch some things off here because I was going to say it a different way, and the program I'm using to make this podcast is a little different than the one I usually use, and I can't erase it, so you're going to have to look at some scratchy ugliness here, but my apologies. But one of these media is going to be complete media that has everything in it except methionine. Okay, complete media lacking methionine. And then we're going to have complete media lacking methionine, and lacking leucine. And then the last one is going to be complete media lacking all three, methionine, leucine, and uracil. All right. Now this strain here, the HFR, will grow on that because it can make its own methionine, it can make its own leucine, it can make its own uracil. Now over here, F minus, I want to have a before conjugation. Sorry for the bad handwriting. I'm using the mouse pad with my finger, so it doesn't always look as legible as I would like it to. All right, so this is before, even though it looks very archaic, and after. Before, minimal media, this one will not grow because you can't, this cell cannot make any of these. Complete, it will grow. And complete, with streptomycin, it will grow because it is resistant to streptomycin. These down here, before the conjugation, they won't grow because in each of these cases, the cell would either have to have the ability to make methionine or make methionine and leucine or methionine, leucine, and uracil. And this cell just can't do that. Now, let's have conjugation occur. And so I have a little bit more room to talk about the conjugation. What I'm going to do is I'm going to redraw this F minus cell closer to it and make the chromosome a little smaller. It's the same one, I'm just moving it closer. So remember, it is met minus, leucine minus, and uracil minus. Same cell. It's also still streptomycin resistant. So I'll just go put SR, just to remind ourselves. Now, conjugation occurs, and what happens is this DNA is copied, and then one of the strands, after it's been copied, will be fed through here. And it'll loop around, and the first part that's going to come through here is going to be this part. And actually, why don't we scratch that out as best as we can? I'm going to draw it over here because we're going to bring more of this in, not just that part here. All right, so we're going to bring in this part here, and we're going to bring in, and we might as well just show it kind of unraveling here. And we're also going to bring in the MET plus gene. In this drawing, even though it's not to scale, leucine and uracil didn't have time to come in here by the time we stopped the experiment. So after a certain period of time, we take the cells and we place them in the blender to disrupt this sex pilus here. And now what the researchers would do is they would take the cells, because remember in this flask, you're growing both of these right with each other. They first grow it on complete media plus streptomycin. And this will grow. These cells will not. Still after. It doesn't change because these cells over here are still sensitive to streptomycin. These cells are still resistant to it. That way, when we're evaluating the data, none of these cells are around. They're all gone. And the only thing that grows are these cells. Now, what's growing here will be cells that had successful conjugation and had the methionine come through, but it would also contain cells like this that did not go conjugation. So how do we know what we're looking at? Well, we're going to use these three media down here. And so what we do is we plate these cells after they've been exposed to streptomycin to get rid of these HFR strains. Now we plate them on complete media lacking methionine. So the only cells that are going to be able to grow are cells that can make their own methionine, which would include the one we just drew up there, because methionine came through. And so now this cell can make its own methionine. So we know from a mapping perspective that the gene closest to this site here, the F plus sequences, the gene closest to it is the gene that makes methionine. Now these still won't grow because this media would require that the cells are able to make leucine and methionine and leucine, uracil, and methionine. And they just can't do that because they're still mutated for uracil and leucine because we didn't allow the mating to go long enough to allow the leucine and uracil genes to come through. Let's say that we let this experiment go 10 minutes. What if we redid the experiment and we let it go 20 minutes? And let's say, because we're making up the rules here, this is our make-believe experiment here, let's say that's enough to get the methionine and the leucine gene through here. So now when you do that experiment, so let's just go 10 here for 10 minutes, and then this one's going to be 20 minutes, like so. Still, you're going to plate it on complete media with streptomycin to get rid of these pesky guys here. We don't want these contaminating our results. It's going to be able to grow in complete media lacking methionine, and it's going to be able to grow in complete media lacking methionine and leucine, because now after 20 minutes, the genes that have come over, and I'm just going to write it here just for a visual, so to speak, leucine's now come over. So now the cell can make its own leucine. And we also know then that the gene next to methionine is leucine. Now let's say we let it go for 30 minutes. And you redo the experiment. You grow in complete media with streptomycin, again, to get rid of these. And now you grow in complete media lacking methionine, leucine, and uracil. The only cells of this F- strain that can grow are those that have had all three of these genes come through. And for our argument, let's just say that 30 minutes is enough time to allow all those genes to go through. We still have plus all the way through. And now for the first time we'll have a plus here, because now the cells can make their own uracil. And so we've mapped the first three genes. We know in E. coli, if we let this go 100 minutes, I don't have another column, but that's okay. If we let it go 100 minutes, that's enough time for the entire chromosome to be moved over here. Now remember, this cell here doesn't lose it, because it copies it through replication, and just half of it comes over here. And then it recombines to give you the double helix that it would need. So this cell here is fine. It's not like it's devastating this cell. It's just sending a copy over here. So I hope this is a slightly better explanation than what we did in class. If you have any questions about this, please make sure you come see me. If not, I'll see you in class. Bye.